Hello everyone, we are going to talk about the urinary system, part 1, structure and function. The function of kidney is to regulate the total water volume and total solute concentration in water, and also regulate extracellular fluid ion concentrations, and also ensure long-term acid-base balance, and also removal of waste, metabolic waste and toxins and drugs. And also it has endocrine functions to secrete renin for regulation of blood pressure, and erythropoietin for regulation of red blood cell production. And also it has a metabolic function of activating vitamin D, and in case of when you are starved, gluconeogenesis to create the new glucose. Urinary system organs are kidneys, major excretory organ, and then ureter transport urine from kidney to urinary bladder, and urinary bladder is where urine can be stored temporarily, and urethra transport urine out of body. As you see, kidney anatomy is at retroperitoneal. That means they are superior lumbar region outside of the body cavity, and they are around thoracic 12 to lumbar 5, and right kidney are normally a little bit lower than left in a position, and adrenal gland is on top of kidney, each kidney, and as you see this, we have a hilus, is the concave side medial surface of the kidney where ureters or renal blood vessels or lymphatics and nerves can exit or enter at the hilum. And then you have a convex lateral face. Outer layer of kidney a dense fibrous connective tissue, and then you have a fat which pads the kidney for protection, paddy cushion, and then also this fibrous capsule of the outer layer normally prevents the kidney not to have infections to spread. And also renal cortex is a granular appearing surface area where filtration units are existing, which is the glomerulus. And then medulla is a cone-shaped medullary pyramid located, and also they are separated by renal columns. And normally urine is condensed in that medullary pyramid, and then into basically drain into papillae and major calyx, and then into renal pelvis through ureter. And blood nerve supplies normally show that kidney cleans the blood, and it adjusts this composition, and it's very rich in blood supply. As you see, that renal artery normally delivers 25% of the cardiac output to kidney each time, and it basically arterial flow normally and into the kidney, and venous flow out of the kidney normally going through the similar path. And I want to make sure that on the right side of slide you want to understand this path of this blood flow. Functional and structural unit that makes the urine is called the nephron. You have about more than 1 million nephrons per kidney, and they consist of glomerulus, that are the border of capillaries. There is a lot of holes in that endothelium, and so that is very highly porous, that allow filtration formation is possible. And the capsules, Bowman's capsule also is called, it's a cup shape which wraps around that border of capillaries. And capsules are making a parietal layer which makes of a simple squamous epithelium, and visceral layer is that's where podocytes are existing, and the foods are sort of like locking up, so on top of a basement membrane outside of the endothelium of the blood vessel, and that creates like a slit, so where filtration can smaller guys, then the filtrate pore can pass through into the capsular space. Proximal convoluted tubules are closest to Bowman's capsule, and then you got descending tubule, and then ascending tubule, and become distal convoluted tubule, DCT, and then you become collecting duct. And the cells which makes proximal convoluted tubule contains cuboidal cells with a dense microvilli, so that it has a high surface area, and then lots of mitochondria, and its function is reabsorption and secretion. And descending limbs consist of simple squamous epithelium, and then they are normally cuboidal to columnar cells, and ascending tubule as well. And then distal convoluted tubules are cuboidal cells with a very few microvilli, and then its function is a lot more in secretion than absorption. And collecting ducts has two types of cells. One is principal cells, which is very sparse and short microvilli containing cells. They maintain water and sodium balance. And the other cell is intercalated cells. And intercalated cells are cuboidal cells. They are much abundant microvilli, and then they are responsible for maintaining acid-base balance of the blood. About 85% of the nephrons exist at the cortical nephron, and then rest is junctal medullary nephron, which is a little bit close to the medullary junction, and that has a lot longer roof, so it invaded deeply into medulla. And so here, ascending limbs have a very thick and thin segments. They are very important in the production of concentrated urine. And the capillary beds are associated with these renal tubules. One is peritubular capillaries for cortical nephrons, and juxtamedullary nephrons are associated with the vasa recta. Glomerulus are very specialized for filtration, and they are fed by arterioles. The one which into the glomerulus is called the afferent arteriole, and once exit out of the glomerulus is efferent arteriole. Normally, afferent arteriole is a little bigger size than efferent arteriole, creating you have some pressure inside, which is very important for pushing the filtrate from the blood out into the cup. Let's see. Vasa recta is very important for more concentrated urine, since it is going into the lot deeper into medulla area. Juxtaglomerular complex are very important for maintaining the pressure within each nephron. And as you see, that they are made of macular denser cells in the afferent arterial wall, and then you have a granular cells within the part of the ascending loop of Henry, and then you got mesangial cells, which is within those glomerulus. So how they actually regulate the rate of the filtration formation and blood pressure is that if macular denser cells sees that a lot of solutes are passing through it, that means that afferent arteriole is too big, so too much blood pressure, so that blood flows a lot quicker into the glomerulus, so they pushes out a lot more stuff faster, so that it cannot be reabsorbed back to blood. Therefore, then macular denser cells will say to JG cells in afferent arteriole, come on, why don't you squeeze a little bit so that less blood can come in. So this is how basically, and opposite is true. If it's too little coming on, that means afferent arteriole is constricted too much, so that blood comes in a lot slower. So this is why that most of the things in the filtrate we observed back to blood. Therefore, they talk with each other to control this net filtration rate. How urine is formed at the nephron is basically about 180 liter of the blood is processed daily into the system, and only like less than 1% become urine compared to the original filtrate. So there are three processes for adjusting this enormous amount of concentration of the filtrate from about 99% back to blood from the filtrate by glomerular filtration and tubular reabsorption and tubular secretion. So filtration produces the cells and then protein-free filtrate. Tubular reabsorption normally selectively returns 99% of the substances from filtrate to blood in renal tubules and collecting ducts. And tubular secretion selectively moves substances from blood to filtrate in renal tubules and collecting ducts as well. And kidney filters body's entire plasma volume almost 60 times each day, and this is very important. The filtration membrane is very porous membrane because as you see this, the podocytes foods can interlock each other, creating little pores in between, so that the water and solutes smaller than the size of little hole can be pushed out from the plasma. So, but normally no blood cells, white blood cells, red cells should be present in a filtrate. So basically the filtrate membrane are made of fenestrated endothelium of glomerular capillaries, and then you have a basement membrane, and then also foot of these podocytes they are locking together to creating this filtration slit. So basically the ones which can push out can be water, glucose, amino acid, urea, and normally bigger proteins like albumin can be cannot moved out of the space, so they retain in the blood. So if you find the albumin in the urine, then something wrong with this filtration or the pressure. So basically the forces that determines the net filtration pressure is determined by outward pressure, which is the hydrostatic pressure. That means the blood pressure, moving pressure, which is about 55 millimeter mercury, and then inward pressure, which is opposing to that force. So that makes it block the filtration basically. So that is a hydrostatic pressure in capsular space, and also osmotic pressure in the capillaries. Those things are inward pressure. So normally you create about net filtration pressure of 10 millimeter mercury, so that stuff can push out of the blood into the cup. So you make sure this nephron try to create enough pressure from the afferent arteriole, having the bigger size compared to the efferent arteriole. As you see, size are a little smaller, creating this pressure possible, so filtration can happen. As you see that nephrons, they love to keep this glomerular filtration rate constant, even if your systemic blood pressure is up and down, because otherwise blood cannot be cleansed or cleaned, right, or filtered. So in this diagram shows that what if that glomerular filtration rates goes down in a nephron because the systemic blood pressure is down. So that means that, as I talked earlier, that sodium chloride in the ascending root of Henle is going to be a lot less than normal, so that macular denser cells will talk with the JG cells in the afferent arteriole. Come on, you need to dilate it so that more blood can come in, so pressure can go up. So this is how glomerular filtration rate can go up. And at the same time, as you see that since the pressure is down, then basically they will try to make the afferent arterial dilation to occur. So that's how they communicate with each other. So this is the intrinsic mechanism within the kidney. However, extrinsic, outside of kidney, can also help try to actually assist to have a constant glomerular filtration rate by using hormonal mechanism of renin, angiotensin, and aldosterone mechanism we talked about earlier, and also using the neural control of sympathetic nervous system is on, so that all the systemic blood vessels will squeeze, so they will create the blood pressure to go up. And also, as you see, the blood pressure is down, and then JG cells and afferent arteriole will release the hormone renin, and that will in the end activate angiotensin 2, and angiotensin 2 will have a effect on vasoconstriction of the systemic arteries, just as sympathetic epinephrine, okay? And also it also has effects on adrenal cortex to secrete the aldosterone, so the aldosterone will work on kidney tubules to have more sodium reabsorption, so water follows into the blood, so that blood volume will go up, so that creates the systemic blood pressure go up. So they work together intrinsically within the kidney, and then also extrinsically outside of kidney try to keep this glomerular filtration to be constant. As you see that most of the tubular contents are reabsorbed to blood through selective either active or passive tubular reabsorption. As you see that they use the two types of the route. One is the passing through the cell called transcellular route. It goes through the apical membrane, into the cytosol, into the basolateral part of the tubule, and into the tissue fluid, and then end up in a peritubular capillaries, so in the blood, and then or between the cells, paracellular. And so those kind of way that water ions such as calcium ion, magnesium ion, potassium ion, etc. can be actually reabsorbed from the filtrate to blood. And as you see, this other organic molecules such as glucose, amino acids, some ions, and vitamins can come in by using the sodium ion as a gradient, as a symphoter, into the cytosol of the tubule cells, and then exit out by using sodium-potassium pump using the ATP. So sodium is reabsorbed back to blood. So that normally the sodiums in a filtrate always end up in the blood. And also water comes in as a what? From the filtrate back to blood as what? As osmosis mechanism. And also fast-soluble substances are very easy to pass the membrane of the tubule cells and end up in the blood. And various of other ions and the urea can come through paracellular between the cells back to blood as well. This shows you the summary of what's happening in a proximal convoluted tubule, and I showed you molecular-wise how it shows. So this is a summary, and this also shows in a descending limb of a U-tube, and where the most of the osmosis of water occurs, and then ascending limb is where basically sodium ions are pumped out, including other ions as well, but major is a sodium ion, so that filtrate is becoming a lot concentrated as it goes down and up. And this also shows you distal tubules, what happened with ions, and also water and urea, and bicarbonate ion as well. And this slide shows you tubular secretion parts, and then reabsorption parts are also summarized here, as I showed a few slides previously. And here secretion parts occurs in a proximal tubule. Hydrogen ions or ammonia has to be secreted out from the blood to the tubule if it's not filtered out, and some drugs you took it, which is a foreign substance, has to be excreted out. And also, as you see this, potassium ions, and then in a distal tubule, and then urea can be also secreted out from the kidney tubules back to the either ascending tubule or to the collecting duct. And as you see this, when you secrete the potassium out, you need the aldosterone to be there. We already talked about earlier. And also, normally that, so secretion is completed right here. So blood can be also, reabsorption and secretion can be complete up to this level, which is the collecting duct. This slide shows you how basically kidney is able to actually concentrate the this counter-current exchange mechanism. So basically what it does is the blood flows in ascending and descending limb of vasa recta, and then filter rate goes to opposite direction to create the basically the best osmotic gradient. We talked about in a class. In the upper parts of the kidney is the less salty, about 300 milliosmol, and as it goes through the helix of the kidney, Magellan helix, it becomes a lot more concentrated. So as you show in the slide right here, top right corner, so top cortex is about 300 milliosmol, so it's a lot less salty, and as it goes down to the tubule, it becomes a lot more concentrated, about 1200 milliosmol. So water is out to descending tubule, and then ascending tubule, sodium is out. So you are making this very concentrated as it goes down, so that you are losing most of the water. So this is how urine is being concentrated. Normally that diluted urine versus concentrated urine occurs. Normally osmotic gradient is normally about 300 milliosmol. So the normal condition urine concentration is a little bit about 100 milliosmol. However, when you are overhydrated, if you are dehydrated, as you see, you will secrete a very little amount of urine, which is concentrated because antidiuretic hormone will be released, and then basically urine will be a lot more concentrated, since most of the water will be reabsorbed with the sodium as well. This slide shows you about abnormal urine constitute. Basically, doctors will evaluate the, what's in the urine to see whether kidney is functioning right, to show the signs of the disease or accessing the renal function. As you see, one example, if you have glucose, high glucose in the urine, creating that your blood must be loaded with glucose, so that when it filters through, the filtration really the spilt over glucose into the filtrate, and so that those tubule cells cannot pick all this glucose back to blood, because you got too much going on, so it will show up in the urine. That shows the sign of diabetic. So you can read about these things and think about what those substances means and what's wrong with the kidney.